And it's now my pleasure to introduce our next speaker, who is going to be Erika Heinrich here at our UCR School of Medicine, who will be presenting Plasticity in the Reflex Control of Breathing, Impacts of Environmental Exposures and Systemic Inflammation. So Erika, you are on. Thank you very much.
Okay, thank you very much for the invitation to come speak today. So our group studies something slightly different. So we're interested in the neural control of breathing. But what I'm going to do today is I'm going to show you how some of what Trevor talked about and Hannah and Talissa as well may yield some unanswered questions in how lung inflammation may impact the neural control of breathing. And there's a lot of unanswered questions here. But I'm going to tell you this story in the context of some of our high altitude physiology work and then bring it back around to inflammation. So before I get into it, just a brief overview of how your body controls breathing. So your body responds to changes in CO2 and oxygen in your blood. So with regards to the response to CO2 or the hypercapnic ventilatory response, you have central chemosensory cells in the brainstem that will respond to changes in CO2 via its effect on the local tissue pH. And then you have peripheral chemoreceptor cells that will respond to decreases in pH. And basically what this entails is that when CO2 in the body builds up, you want to get rid of it. And so you'll increase your breathing to do so. And so as the PCO2 in the blood increases, you reach a point at which this is called the ventilatory recruitment threshold. And above this point, your breathing will start to increase to get rid of this extra CO2. You also have something called a hypoxic ventilatory response. So as oxygen starts to fall in the blood, these peripheral chemosensory cells will start to increase breathing as a function of the PO2 in the blood. So these are the two breathing reflexes that we measure in our lab. And you would think that these two reflexes, since they're sort of the body's first line of defense to defend against hypoxia and the pH changes in the blood, you would think that they'd be very conserved across individuals. But in fact, they differ significantly both across human populations as well as within individuals, depending on different environmental exposures or different disease states. So for example, we're very interested in the impact of chronic hypoxia exposure on the control of breathing. So for example, at high altitude, as you increase in elevation, the partial pressure of oxygen in the inspired air decreases. And so if you were to take someone who's a sea level resident and bring them up to high altitude, so 3,800 meters, and allow them to acclimatize, this hypoxic ventilatory response, or the increase in breathing as oxygen in the blood decreases, it increases. So you're more sensitive. But if you look at what happens in people who live chronically at high altitude, so for example, native populations in the Peruvian Andes, if we do the exact same test in that population, they have almost no ventilatory reflex to hypoxia. It's really amazing. But clearly you can see that this reflex is highly plastic. And what we see in the Andean population is not true of all high altitude groups. So for example, people who are native to the Tibetan Plateau tend to maintain much higher resting ventilation and higher hypoxic chemosensitivity, or their breathing reflex to oxygen. So you can see in the Andean group, they have a lower mean HVR and resting ventilation, and they have none of these high responders. So again, this is a very plastic reflex that changes with environmental exposure, genetic predispositions, and across populations. And it also changes significantly in different disease states. And I'm just going to show this data for asthma here. So these are asthmatic adults versus normal healthy adults. And basically what these plots are showing is that the ventilatory sensitivity to hypoxia starts to become blunted in people with chronic asthma. So you can see here, basically as you go right on the x-axis, that's decreasing oxygen in the blood. And so you get a higher increase in ventilation in a normal person versus someone who has chronic asthma. And the respiratory drive, or sort of how forceful their inhales are, is also elevated in someone who's a healthy, normal control. So the reason this is important in terms of sort of clinical significance is that if you have high respiratory drive, it can be protective against things like obstructive sleep apnea, or it protects you against hypoxemia in the context of lung disease. But it can also predispose you for central sleep apnea and self-inflicted lung injury during critical lung disease. And someone with high respiratory drive also tends to feel more short of breath. So for example, a child with asthma, if they have high respiratory drive, they'll sort of feel more short of breath during an exacerbation than a child with a lower respiratory drive. So on the other hand, someone with low respiratory drive, like I said, may feel less short of breath, but they'll be protected against self-inflicted lung injury. But this can lead to worsened obstructive sleep apnea and worsened hypoxemia in lung disease. So these reflexes are very important for sort of downstream clinical or disease development. So we've been interested in what role systemic inflammation may play in modulating these reflexes. And there's two reasons we were interested in this. First of all, we study, how hypoxia can impact inflammation. And I won't go into detail here, but this figure on the right is just showing that sort of these molecular pathways that control the response to oxygen are very connected to the molecular pathways that control the response to infection and inflammation. So these are two very tightly linked signaling mechanisms. Also, we know that in many diseases that are associated with chronic systemic inflammation, there are significant changes in how these breathing reflexes manifest. So in obstructive sleep apnea, you often get elevations of the respiratory drive. In asthma, you can get blunting of the respiratory drive. And in COPD, it can sort of go either way. And then in a disease called chronic mountain sickness, we get blunting of the respiratory drive. So to determine what role or what link there may be between systemic inflammation and the ventilatory reflexes, we did an experiment using our high altitude exposure model where we recruited 15 healthy sea level residents, and we collected physiological parameters in blood at sea level, and then over 3 days of acclimatization at 3,800 meters. And maybe some of you in the audience may be familiar. This is Barcroft Station. So it's up in the Eastern Sierras, and it's part of the UC Natural Reserve System. So we get to stay in these luxury accommodations for several days. And we also measured ventilatory reflexes at sea level and after 2 days of acclimatization. So the first thing we did was we looked in the blood, and we wanted to see whether or not hypoxia, in the absence of any other infection, created inflammation, and it did. It created a huge systemic inflammatory response. So these plots on the right are basically just showing when you go to high altitude, a lot of genes are differentially expressed. But if we take those differentially expressed genes and we ask, what are the biological pathways that they're enriched for? We see things, these are the top pathways, and we see things like positive regulation of IL-8 production, toll-like receptor signaling, interleukin-6 production. So these are all pathways that are sort of priming the innate immune system and creating systemic inflammation. And this is probably kicked off by the increase in some of these alarmin molecules. So we saw these screaming signals where HMGB1 and several of the S100 proteins, which signal tissue damage, they were hugely increased at high altitude compared to the sea level values. And this is important for the priming of the innate immune system, sort of like what Trevor was getting at in some of his work. So we saw that many factors involved in TLR4 or toll-like receptor 4 signaling. This is a pathway that helps you respond to a bacterial infection. And many of these factors were also upregulated at high altitude. Again, despite having no secondary infection in any of these people. So the TLR4 receptor itself was upregulated. We also saw upregulation of this MD2, which is a co-receptor that helps bacterial LPS bind. And CD14, another co-receptor, was also upregulated. And then, of course, HMGB1, which is this alarmin. It says the tissue is hurt, we need to respond. That molecule can also signal through this pathway. And you can see that when this becomes activated, you get an increase in pro-inflammatory cytokine production. So our question was, could this systemic inflammatory response be linked to the increase in hypoxic ventilatory drive that we see at high altitude? Because we know that things like sepsis, LPS stimulus, or sustained or intermittent hypoxia can lead to systemic inflammation that can modulate the function of these chemosensory cells, which are sort of what tells your body to increase breathing. And so that can lead to downstream impacts on the control of breathing, regulation of the heart rate and blood pressure, and renal function. Furthermore, if you treat either human participants or rat model with ibuprofen before you expose them to hypoxia, you can blunt this increase in respiratory drive. So clearly, inflammation at the systemic level plays a role in the modulation of these breathing reflexes. So we measured these reflexes in these participants. Just a quick old photo of me doing one of these tests. We basically have people breathe in and out of this bag, which allows their CO2 to build up over time. And we do that 2 times at 2 different oxygen tensions, one high and one low. So you can see as the CO2 increases while they're rebreathing, you get an increase in ventilation here. And if we pull out that data, it gives us something like this, like I showed you early on, where you have this sort of hockey stick shape where you get an increase in ventilation as the PCO2 increases. So we did see that several of the inflammatory markers that were increased at high altitude were associated with higher CO2 gain. So I'm just showing you the CO2 response here, as well as a decrease in the ventilatory recruitment threshold. This is amazing because what this shows is that someone with lower inflammation would look more like this blue line. So their threshold is shifted right and they have a lower slope of this response. Whereas someone with higher levels of inflammatory biomarker expression, this threshold point is shifted to the left and the slope is higher. So who cares? Well, this means that for 2 individuals with different levels of systemic inflammation, if they have the same CO2 in their blood, one person is breathing a lot harder than the other person. And so that can have implications, like I said, in an asthma exacerbation per se. Someone may feel much more short of breath, much more panicked than another, and they may be at a higher risk for self-inflicted lung injury. So I'll just finish up by tying this back to sort of this air pollution topic and sort of ties into the fire smoke exposures as well. So we already know that chronic air pollution exposure can lead to systemic inflammation, which can have downstream implications for vascular function, atherosclerosis, and other autonomic functions. But there's really not much known about how chronic air pollution exposure impacts the neural control of breathing. So this is a really interesting and fruitful area where there's a lot of questions left to be answered. So if there's anyone interested in this topic, I welcome you to reach out to me. And it's been really great to hear from everyone else in the group today. So just a couple acknowledgements for the people who helped collect this data, and I'd love to take any questions.
Thank you, Dr. Heinrich. That was awesome. And you did such a great job of being on time, in fact, a little early, that we can definitely take a moment now to take any questions if someone would like to enter any into the chat box.
It always takes me a while to type, but I can keep an eye on it as we go.
I'll ask a question while folks may be getting their questions into the chat box. We've heard, of course, members of the group talk about work where they're looking at mouse chamber exposures just now, as well as the microbiome differences among particles in the Salton Sea area. I was curious about connections that may be between your work and these kinds of topics. If you had any kinds of interesting areas this could overlap and have that Venn diagram.
So something that I think is pretty important, and this is going to be, you know, a long-term issue, is if we have people growing up with these chronic exposures to high levels of air pollution, we don't really have the data yet, but for the control of breathing, but we do know that these chronic exposures are impacting other cardiovascular functions. But when you have someone who starts to develop COPD or chronic asthma, or COVID-19 happens, you know, the way that you respond to that acute disease or to that chronic disease is very closely tied to your respiratory control. Because if you have blunted respiratory control, you're going to have lower oxygen availability in your blood, which is going to lead to a whole slew of downstream effects. And so I think the control of breathing is a really important aspect of this whole story that we don't really know a lot about. And, you know, I don't know if we need longitudinal studies to look at some of this, but the exposure chambers would be a really good way to start looking at this.
To your knowledge, are there any such longitudinal studies being planned, or is that something that at some point you would be interested in spearheading?
I don't know of any, you know, with regards to particulate matter exposure. It's sort of funny because I have seen, you know, studies get IRB approved to expose people to particulate matter, human subjects. And so these are experiments that are possible for acute exposures. But yeah, I don't know. It would be really interesting to look though.
Excellent. Thank you so much, Dr. Heinrich. And